being here in Zaragoza lecturing to the MSc course in animal nutrition is a very exciting time for me because it gives me an opportunity in my lectures, which are on nutrition and reproduction and pregnancy and lactation and farm animals. It gives me an opportunity to talk about one of my favorite subjects, epigenetics. This is a new science, and one in which we got into quite by accident, really. I was interested in fetal growth and development and nutrition, how it affected the growth of the fetus. And when we described that mathematically using sophisticated mathematical equations and back extrapolated those equations, which had very fundamental biological meaning in all of their coefficients, we found that we literally just went through about the origin, indicating that something was happening very early in the fetal growth processes and development processes that impinged on the later development and birth of the newborn. And then, of course, we also got involved with the in vitro culture of embryos and the background media in which they were in. And we found that in some of these conditions, we got alteration in the expression of genes. And this really led us into the field of epigenetics because we hadn't changed the genetic material. Epigenetics doesn't change the DNA. The DNA stays the same. But as a result of the background environment that those early embryos are exposed to, the nutrient supply that they see in their environment that they're growing in, alters the degree to which those early genes in the first few days after fertilization are methylated and thus expressed in later life. So at that particular point, we find that something, for example, like in the case of an animal cobalt deficiency, in the case of a ruminant, which leads to vitamin B12 deficiency, because ruminants normally supply their own B vitamins, B12 included, from rumen microbes, but they need for B12 the cobalt. But when we found that we had subclinically cobalt deficient animals, subclinically B12 deficient, they look perfectly healthy to the farmer, but in actual fact, the viability of their newborn lambs was adversely affected by the lack of methylation of important genes that depended on B12 at those early stages of embryo development. They require the B12 in the methylation process or the expression of the genes. What we are finding now that in those particular experiments, these animals had higher, as adults, blood pressure, they had more prone towards diabetes, they were less sensitive to insulin, they were unable to mount as good an immune response when challenged with an antigen or with a disease. And they were less muscular, which is obviously important from a farming point of view. But all these health and production traits were influenced in the first 6 days of early embryo development. The genes had not been correctly activated to give optimum health and performance thereafter. And I think what we're seeing now in this exciting new field is tremendous opportunities for you students that are coming here to study and do projects in research in animal nutrition is to look at specific nutrients that are involved in these methylation processes. More of these are coming online each day. We know, for example, choline is another methyl donor, methionine is a methyl donor. And if our diets are low in these, and now we are beginning to realize that in humans, quite a few of these situations arise where diets are inadequate, perhaps to give full methylation to our genes. So the animal studies are beginning to, in fact, leading the way in many ways into medical studies that affect our health as well. So I think it's tremendous opportunity to get in to this exciting field. I've just quoted one or two examples that we personally have been involved in experimenting with, but I think there are many opportunities for you to look at other specific nutrients, particularly the vitamins. We have known about folic acid and neural tube defects and what have you, spina bifida in the past, but this has opened up a whole new area of control. Now, it is an area which we thought you could do nothing, environment couldn't alter the expressions of genes. Environment does alter the expressions of genes, and it is there to be carried through to subsequent generations as well, which means it's something we cannot ignore. It is important not only to our children, but to our children's children.